Okay, let's dive right into this incredibly exciting explainer. Today, we're looking at the superpowered microscopic machinery that is literally keeping you alive this very second. We're going to explore the fascinating world of enzyme kinetics and uncover the hidden mechanics of biology that make life itself possible. Seriously, you are about to see just how brilliantly engineered your own cells really are. You might not realize it, but inside your cells right now, there is a ticking biological clock. And without a secret weapon, well, you simply wouldn't survive. Think about it for a second. Your body is essentially a giant walking chemistry set. Every single second, millions of reactions need to happen just for you to digest your food, move your muscles, or even think the thoughts you're having right now. But here's the catch. Left on their own, many of these chemical reactions are far too slow or just too energetically unfavorable to happen on a biological timescale. So how on earth does your body pull off this incredible feat? Well, the answer lies in a number that is just mind-boggling: 100 billion. That's a 1 with 11 zeros. We have these mighty molecular machines that speed up biological reactions by up to 10 to the 11th power. An enzyme-controlled reaction moves so much faster than a non-enzymatic one that a chemical process that might naturally take years can actually be knocked out in mere fractions of a second. Section 1. The molecular speed machines. Let's formally meet these biological speed racers known as enzymes. This group includes catalytically active proteins and occasionally RNA molecules known as ribozymes. They are the driving force behind almost every single type of biochemical reaction related to your cell metabolism. To really grasp this need for speed, let's look at a specific enzyme called carbonic anhydrase. This little guy stops a dangerous cellular buildup of carbon dioxide by dramatically accelerating how fast it dissolves in water. The real-world application here is super fascinating. Carbon dioxide is a waste product of your cellular metabolism, right? It has to be cleared out of your cells and transported into your bloodstream so you can exhale it. The problem is that CO2 dissolves in water very, very slowly. If we had to just sit around and wait for it to happen naturally, we'd be in big, big trouble. But enter our hero, carbonic anhydrase. A single molecule of this amazing enzyme processes 600,000 molecules of carbon dioxide per second. It increases the reaction rate by a mind-blowing factor of 10 million. Just one tiny protein molecule is doing all that heavy lifting, turning what would be a sluggish chemical process into an absolute blur of biological efficiency. Which brings us to Section 2, crushing the energy barrier. So you're probably wondering, how do enzymes actually pull off this metabolic magic trick? How do they make the impossible happen so quickly? Well, it all comes down to breaking through energetic barriers. To get from a starting substrate to a final product, molecules naturally have to climb a massive energy mountain. That's what we call the activation energy. But enzymes basically act like a shortcut tunnel, instantly lowering that daunting mountain into a tiny molehill. By stabilizing what's called the transition state, they allow the chemicals to easily and rapidly cross over to become products. And honestly, the absolute best part is that the enzyme acts as the ultimate reusable helper. Because it's a true biological catalyst, it emerges completely unchanged and totally ready to get right back to work at the end of the reaction pathway. It doesn't actually alter the reaction equilibrium. It just gets you to the finish line much, much faster. One single enzyme can perform its function over and over again, thousands of times a second. Moving right along to Section 3, the enzyme naming code. With thousands of different machines to keep track of, scientists actually came up with a pretty logical cheat code for naming them. Identifying an enzyme is basically like reading a secret language, where the name almost always ends in ace and tells you exactly what its job is. For example, if you hear the word sucrase, you know right away it reacts with sucrose. If you hear lipase, it works on lipids. Sometimes the name just describes the function outright, like an oxidase that catalyzes an oxidation reaction. Now, of course, there are always a few classic exceptions from the early days of biology, like our digestion enzymes pepsin and trypsin, which stubbornly end in "in." To keep the whole shebang beautifully organized, all enzymes fall into one of 6 neat classes based entirely on the specific type of chemical reaction they conduct. We've got oxidoreductases that handle oxidation and reduction, transferases, which, you guessed it, transfer groups of atoms around, hydrolases break bonds using water in a process called hydrolysis, lyases add or remove atoms to double bonds, isomerases shuffle atoms around to rearrange a molecule, and finally ligases are the builders that combine molecules together using cellular energy or ATP. All right, let's zoom in for section 4, inside the active site. We know what enzymes do, but where exactly does this high-speed chemical handshake take place? Well, the magic happens at the active site. Enzymes themselves are large, complex, three-dimensional globular proteins, but the active site is actually a relatively small region. It's this tiny, highly specific pocket made of just 3 to 5 amino acid residues. Here, the substrate, which is usually a much smaller molecule, gets perfectly hugged by multiple weak, non-covalent bonds. The specific shape and the unique chemical environment inside this little pocket are exactly what permit the chemical reaction to proceed so effortlessly. It's basically a beautiful 3-step dance, which we can see perfectly during the hydrolysis of sucrose. First up, binding. The substrate sucrose specifically binds to the active site, forming what we call the enzyme-substrate complex. Second is catalysis. The sucrase enzyme helps break the chemical bond using water. And third, dissociation. The newly formed products, glucose and fructose, are released, and the sucrase enzyme resets, perfectly intact and ready to grab another sucrose molecule. Now, for a long time, scientists thought of this active site interaction as a super rigid lock-and-key situation, where the enzyme is a stiff lock and the substrate is a perfectly shaped key. But we now know it's a bit more dynamic than that. It's actually an induced fit. Think of it more like a flexible glove gently molding around your hand for a perfect fit. Both the active site and the substrate slightly adjust their shapes as they come together, just to ensure an exact, snug alignment for the reaction. And that brings us to section 5, helpers and coenzymes. Look, even heroic, highly specific enzymes sometimes need a little help from their microscopic friends to get the job done. Not all enzymes can work entirely on their own just using their protein structure. They rely on non-protein helpers known as cofactors, which come in 2 main varieties. First, you've got essential mineral ions, things like iron, zinc, copper, and magnesium, which help with substrate binding and catalysis. Second, you have coenzymes, which are organic compounds. And here is a really fun fact for you. Many of the coenzymes your body relies on are actually derived from the dietary vitamins you eat every single day. This is quite literally why getting your vitamins and minerals is so crucial on a fundamental cellular level. Just to get the terminology straight here, without its trusty sidekick cofactor, the protein portion flying solo is just an inactive apoenzyme. It's built, sure, but it's not quite ready to work. But when they unite, when that apoenzyme binds with its specific metal ion or coenzyme, boom, they become a fully functional biological powerhouse known as a holoenzyme. Remember our super fast friend from earlier, carbonic anhydrase? Well, it's a classic example of this. It's a metalloenzyme that relies entirely on a tiny zinc ion situated right at its reactive center to stabilize its reactions. Without that zinc ion acting as a cofactor, carbonic anhydrase just couldn't do its job, and our blood wouldn't be able to stay perfectly balanced. So, as we wrap up this explainer today, I want to leave you with a fascinating question to chew on. If these speedy, specific, and highly regulated little enzymes are the precise clockwork of life, what happens when they go bad? If they lose their structure, become denatured, or start accelerating the wrong reactions entirely, it disrupts the whole system. In fact, when enzymes misbehave, it can drive severe diseases and even lead to the development of cancers. Understanding enzyme kinetics isn't just about knowing how we function on a good day, it's the absolute key to understanding how to fix the machine when it breaks. Thanks for hanging out and exploring this molecular speedway with me, and definitely keep questioning how the amazing machinery inside of you works.